Very interesting. Okay.
But, thanks very much for the invitation, Alison. I just want to say it's been really infectious, the enthusiasm of the iGen students, and it's wonderful to be somehow associated with the program.
Thank you very much.
So just to get a feel for the audience here, do we have some people who are not biologists? If you put up your hands. Okay. So you're probably not very familiar with protein synthesis, so I might go a little bit slower in those areas, and also I'm happy for interruptions as we go. So what I'd like to do is talk about maybe the first real movement of synthetic biology into drug discovery. It's taken a while, and it's difficult, and we hope some drugs are going to come out of this. So we have two synthetic biology projects in our lab, one of which is focused towards synthesizing a ribosome translation and a minimal cell or life. We think we understand cells and life pretty well, but then when you come to try to put it together from parts, you realize there's a lot we still don't understand and we need to learn. And one of the best ways of learning that is to try to build it. And we've reconstituted protein synthesis. We're working on ribosome RNA modifications and how to put that together into something that can replicate. And this could have new applications in biotechnology. But I don't want to focus on that for this short talk. I'm going to talk about the other project, which is translation with unnatural amino acids for drug discovery. Designing new genetic codes that make peptides containing unnatural amino acids. I see there are a couple of bonds that have disappeared here, but we'll get to that. So, ligand and drug discovery, binder discovery, if you like, small molecule binder discovery, has undergone a revolution in industry over the last decade. And many drug discovery programs start with these $2 million robots that pipette about 1 million different compounds into an assay designed for high throughput screening. And it takes about 2 weeks for this process, and it's extremely expensive. It costs about $30,000 or $40,000 to do one of these screens, apart from purchasing the robot itself. But at the end of the day, one inevitably gets low affinity small molecule hits to any target that one picks. And there begins the process of drug discovery. And it's a very long one from there, because you need to optimize these low affinity hits to get much more higher affinity potent hits to help reduce toxicity and dose issues. But even then, there are many other hurdles once you start going into animal models and humans of metabolism of the drug and unexpected effects. But nevertheless, the amount of work required in chemical synthesis here, which is not done in parallel, and the screening is enormously inefficient and takes a long while to get very potent molecules. Using Darwinian evolution is a faster approach, and many of you are aware that the most successful form of drug discovery today is antibody discovery, if we can call it drugs. Some people don't quite call antibodies drugs because they are sort of biologicals. They're very big polypeptides. You can't inject them. They don't penetrate penetrate cells. You can't swallow them, you have to inject them. They don't penetrate cells. But nevertheless, it is now quite a rapid process to discover very high affinity antibodies using literally evolution, monoclonal antibody technology, evolving antibodies out of a pool of perhaps some 10 to the 16 different molecules for binders that are very specific, very high affinity for their target. And because of this tremendous specificity and potency, quite often they can be drug candidates already. But as I said, they have these problems with pharmacokinetics. So Joyce, Gold and Shostak developed a completely in vitro evolution technology that encompasses the three important steps of Darwinian evolution, which is mutation, which overcomes the diversity limit in your library for selection, selection which overcomes the background limitations in your selections, and amplification which overcomes your sensitivity limits. And so using these three processes reiteratively, one can select from huge pools of RNA molecules, high affinity ligands or aptamers to any particular target of interest. But again, as when you face drug discovery issues, you have the problem of cell impermeability of a highly negatively charged polymer and susceptibility to ribonucleases. So though you can find high affinity ligands very rapidly with RNAs and antibodies, they're not very useful for many of the targets you wish to address. The technology can also work with peptides, and here you can overcome cell permeability with some peptide sequences, but then they're susceptible to protease digestion and rather immunogenic because of the way they bind to MHC molecules. So I started my research in the RNA area, but I moved into the peptide area because I saw the potentials here in drug discovery, and I'd like to